guys, welcome back to my EduSpace. Hope you all are doing and learning well. In the previous video, we talked about the structure of an eukaryotic cell. What are the components that involved in the formation of an eukaryotic cell? We discussed in brief about the cell organelles. In today's video, we will look at the detailed structure of a cell membrane or plasma membrane. So, without taking much time, let's get started. So, what is the cell membrane? The first question that click in our mind is, what is the cell membrane? What is the significance of this membrane? Before answering this, let me tell you one thing, that why we use is homes, because it protects us from the surrounding. Just like that, cell needs some envelope that protects them from the surrounding. So this envelope is known as cell membrane, that separates interior environment of the cell from the external surrounding and thereby protects the cell from the surrounding. So the cell membrane, also known as plasma membrane, is found in prokaryotic as well as eukaryotic cell. Structurally, the cell membrane is similar in prokaryotic as well as eukaryotic cell. Basically, it separates the interior of the cell from the outside environment and protects the cell from the surrounding, as I just told you. The cell membrane consists of a lipid bilayer which is semi-permeable in nature. I hope that you all understand what is the semi-permeability means. Let me tell you what is the mean of semi-permeable. It means selective choosing. It means selective choosing of molecule which can pass through the plasma membrane. Selective selection. Selective selection. Suppose this is our plasma membrane. Okay, this plasma membrane is semi-permeable in nature. It allows only some selective molecules to pass through inside the cell or outside the cell. So, the function of the plasma membrane is to control the movement of ions and molecules in and out of the cell and cell signaling. It also serves as a receptors. Now, one question comes in your mind, that why cell membrane is lipid bilayer? Why two layers of phospholipids are required to form a cell membrane? As we just discussed that it consists of an inner membrane and outer membrane. So understanding this, suppose if you place some phospholipid molecules inside water. This is water, H2O, and you are going to place some phospholipid molecules. As the structure of the phosphomolecule is like this, head and two fatty acid tails. Head is hydrophilic and tails are hydrophobic. So when we place this phospholipid in the water, then you will see it forms a sphere. Okay, they forms a sphere like this. These are the head which pointed toward the water and the tail which always pointed away from the water. So like this. They form the structure like this. That's the case in fats. Okay, if we place a drop of oil in water, it also tend to occupy the sphere in water. Just like that, these phospholipid molecules forms a sphere inside the water where the interior of the sphere is occupied by tail, because these are hydrophobic. The interior of the sphere is occupied by tails and the outer portion of a sphere is occupied by head. Okay, like this. But in case of a cell, okay, but in case of a cell, the cell interior is cytoplasm, which is aqueous, and the outer surrounding is also aqueous, that is extracellular matrix. So tails tend to come together as they are hydrophobic and head pointed toward the aqueous region. Thereby, these phospholipid molecules form bilayer. Let me explain you with a diagram. Suppose this is a cell, okay, and this is the extracellular matrix and this is the cytoplasm. These are the cytoplasm. Outside aqueous region, extracellular space is also aqueous and cytoplasm is also aqueous. So these molecules tends to arrange in this behavior like this. And so on, okay, and so on. So the head pointing toward the extracellular space forms outer membrane, okay, and these phospholipid molecules where heads pointing toward the cytoplasm forms inner membranes, and both the tails of outer and inner membranes tends to occupy the interior portion which forms the hydrophobic core of a plasma membrane. Hydrophobic core, because it's make it's made up from these fatty acids chain which are hydrophobic in nature. Now the structure of cell membrane. The cell membrane is composed of phospholipid molecule. This is the molecule which make the complete plasma membrane. Beside this phospholipid molecules, there are some proteins which also help in the formation of the plasma membrane. So first let me discuss about the phospholipid molecules. What is this? Actually, two layers that is formed by this phospholipid molecule is required for the formation of a plasma membrane. This is the plasma membrane you can see over here. This is the phospholipid molecule, okay. So we require two phospholipid layer to form the plasma membrane. The outer one, which faces toward the extracellular space, is known as outer membrane, and the inner one, which faces towards the interior of the cell or at the cytoplasm, is known as inner membrane. So two phospholipid layers are required. That's why it is known as phospholipid bilayer or lipid bilayer. So cell membrane consists of a phospholipid bilayer, that means two layers of phospholipids which make the inner membrane and outer membrane. So this one is the outer membrane because it faces towards the extracellular space, and this one is the inner membrane because it faces towards the interior of the cell, or we can say at the cytoplasm. A phospholipid molecule is having a polar phosphate group and two non-polar fatty acids and a glycerol group. Okay, it consists of a polar phosphate group, two non-polar fatty acids and a glycerol group. This phosphate group forms the polar head. It forms the polar head and this forms the non-polar tail. Okay. The phosphate group which forms the polar head is hydrophilic in nature. This one is hydrophilic in nature. Hydrophilic means which loves water. Hydrophilic in nature. So, as it is hydrophilic in nature, so it is obvious that it hates lipid. So it is lipophobic. Okay, it is lipophobic. Lipophobic. So it consists of a head and tail. The head is polar. Polar means hydrophilic or lipophobic. Okay. So if heads are hydrophilic, so it is obvious that this is lipophobic, which means lipid haters, as the polar molecule could not mix with oil or fats. So the head part of the phospholipids always points towards the aqueous region. You can see here the head portion is always faces towards the aqueous region, whether it is extracellular space or at the cytoplasm. Now the two fatty acids chains forms tail, this tail, and these are hydrophobic in nature. So we can say these are lipophilic. They have attraction towards the fats. These are lipophilic. And they always pointed away from the aqueous region. These are the tails. You can see these are the tails and these are the tails. Where they are facing? Are they facing to the aqueous region? No, they are facing away from the aqueous region. So for this particular molecule, we can look here, and for this particular molecules, the head is facing toward the aqueous region and the tail away from this. Same like that, in this case of molecule, the head is facing toward the aqueous region of cytoplasm and the tail is facing inward or away from this region. So basically they form the interior hydrophobic core of the plasma membrane. This particular region is known as the hydrophobic core of plasma membrane. Pictorially, the phospholipid molecule can be represented as this, the head and two tail. So in this picture of cell membrane, you can see over here the cell membrane is having two layers. The outer one is outer membrane and the inner one is inner membrane. They forms the phospholipid bilayer. The inner membrane has hydrophilic head pointing towards cytosol and hydrophilic tail pointing away from the cytosol. The outer membrane has hydrophilic heads pointing towards the extracellular space and hydrophobic tail pointing inwards away from the extracellular space. I hope you understand the structure, why it is lipid bilayer. So next one is, yes, beside phospholipids, the cell membrane also composed of proteins. So basically two types of proteins are embedded in the cell membrane. These are integral proteins and peripheral proteins. So look at this picture. These are some proteins molecules. You can see in blue color, these are some protein molecules. These are some proteins. Some are tightly binded, or we can say integrated with this phospholipid bilayer, or we can say span the entire membrane. These are integrated protein or integral protein, or we can say intrinsic proteins. Hence, they cross the entire membrane. They act as a transporter. So, these integral proteins act as a transporter proteins. And those proteins which you can see over here, which are not completely span the entire membrane but loosely attached. These are loosely attached on the surface of this bilayer membrane. These proteins are known as peripheral proteins. So, integral proteins, these span throughout the entire membrane and thereby functions in transporting ions and molecules from extracellular space to the cytoplasm or vice versa. This is the integral protein, or we can say intrinsic proteins. They help in transporting the molecules inside or out of the cell. These proteins are known as membrane transporters. So you have to remember this term, the integral proteins which span the entire lipid bilayer is act as a membrane transporters. Okay. Now the peripheral proteins. These proteins do not span the entire region. See, you are saying this is span the complete region of cell membrane? No, these are loosely attached to one layer of plasma membrane. Okay, they bind either to the integral proteins or to the phospholipid head with ionic bonds. So what are the bonds which require them to get in attached with this cell membrane? These are ionic bonds, which we know that these ionic bonds are weaker bonds. These peripheral proteins can bind to either this phospholipid molecules or to the integral proteins. They are also found in attachment with these integral proteins. So functionally, they help in cell adhesion, cell recognition, and cell signaling. So what we have discussed till now, we have discussed about the structure of cell membrane. Beside the phospholipid molecules, there are some proteins which also helps in the formation of plasma membrane, and especially they are useful in the functioning of cell membrane. There are two types of proteins. The first one is integral proteins or intrinsic proteins, this, and the other one is extrinsic protein, or we can say peripheral proteins. You can see over here we have three types of integral proteins. Some are alpha helix protein, okay, some are integral globular protein, and some are protein channels. These are protein channels. The alpha helix protein is the major category of transmembrane proteins, and most of the helical domains, this helical domains, these are hydrophobic in nature, or we can say non-polar, thereby span the hydrophobic region of the lipid bilayer. Next, the globular protein. These are also known as spheroproteins. These are known as spheroproteins. Spheroproteins. Why these called spheroproteins? Because these are spherical or globular in shape. They are very common in plasma membrane, and these are polar in nature, which helps in transport, cell signaling, or catalysis, that is enzymatic activity. Now this channel protein, as its name suggests, that they forms a channel which allows incoming or outgoing of ions or molecules. Channel proteins span the entire membrane and make hydrophilic tunnel. Okay, the way that they form is known as hydrophilic tunnel across the cell membrane. This channel are very selective and allow only one type of molecule to pass through them with the help of diffusion process. Now next component is glycoproteins. You can see over here there are some oligosaccharides chain. Here green, this blue, green. Okay. These oligosaccharide chains are attached on the integral protein, somewhere on the integral protein like this, and as well as on the peripheral protein. This is the peripheral protein where the oligosaccharide chain you can see in blue color is attached, which are held together by strong covalent bonds. Such carbohydrate and protein complex is known as glycoprotein. This is the complex which forms from the oligosaccharide chain and a protein. So this complex is known as glycoprotein. This carbohydrate chain usually protruded outside the cell. You can see this is protrude. Most of the oligosaccharide chains are protruded outside the cell in the extracellular fluid. Example includes collagen, which is a structural protein, mucin, which produce lubricants, transferrin, which is transport protein, and immunoglobulins or our antibodies. Now, what is fluid mosaic model? This biological model was devised by S.J. Singer and Jill Nicholson in 1972, which states that this is not an intact membrane. Okay, this is not an intact membrane like paper or polythene, but all the molecules which forms the plasma membrane like phospholipid, okay, cholesterol, these are the cholesterol molecules, okay, and the proteins, this, they are constantly moving, they are not static or stable structure. Okay, they are always tends to move laterally here and there. With the movement of these phospholipid molecules, the intrinsic proteins as well as the peripheral proteins and the cholesterol, they also moves constantly. This movement is due to the sideways or lateral movement of phospholipid molecule that we will see in our upcoming videos. This is the phospholipid molecules and it has the property to always move laterally, either to the left or to the right. So there is a always sideways movement. Due to this complete membrane, cell membrane included these proteins and cholesterol also constantly moving. So With the continuous movement of phospholipid molecules, the cholesterol and proteins that two attached with them are to move. So proteins and phospholipid molecules are bind together with intermolecular and electric forces. Due to these weaker forces, lipid and proteins do not remain at stationary, but they constantly move in sideways direction. This is reason behind the fluid mosaic model. Now the last but not the least is cholesterol. It comprises 30% of lipid content in cell membrane and extremely important component. Okay, cholesterol. From all the lipid content of the plasma membrane, 30% is from this cholesterol. Functionally, it acts as a regulator of membrane fluidity. It regulates the membrane fluidity at various temperatures. At high temperature, it stabilizes membrane and raises its melting point, and at low temperature it intercalates between the phospholipids and prevents them from clustering together and stiffening. So at high temperature, when we raise the temperature, suppose these are the phospholipid molecules. Okay, you can say cholesterol also act as a buffer. These are the phospholipid molecules which forming cell membrane. And when we raises the temperature, if the temperature raises, what they does, their movement increases and they tries to go far away, which increases or raises the fluidity of the cell membrane. Okay, but the cholesterol which are present inside this, like this, they stabilizes the membrane and raise the melting point. It raised the melting point. So with increasing temperature, they could not go much far away and stabilizes the fluidity of membrane. On the other hand, when at the low temperature, they try to come very closer like this in the absence of cholesterol, and they could get stiff together, due to which the membrane fluidity will stops. So with the presence of the cholesterol, they could not come as much closer together or cluster together, because these cholesterol molecules occupy some space, and due to this space they are having difficulty to come closer and stiffening. So cholesterol is very important for the fluidity in plasma membrane. Thanks for watching.